Okay, I think we can go ahead and get started. So good afternoon, everyone. It's my pleasure to introduce today's speaker, Dr. Carrie Shawber, Associate Professor of Reproductive Sciences in the Departments of Obstetrics and Gynecology and Surgery at Columbia University. Dr. Shawber received her BS in biology and PhD in molecular biology from UCLA, followed by a postdoctoral training in cancer genetics and vascular biology at Cornell and Columbia. Dr. Shawber is an internationally recognized vascular biologist whose research focuses on the development and dysfunction of the lymphatic system. Her laboratory investigates the genetic and molecular mechanisms underlying lymphatic disorders, including lymphatic malformations, congenital chylothorax, and generalized lymphatic anomalies. Her work has been supported by the NIH, the DoD, and multiple private foundations, and has contributed significantly to our understanding of Notch signaling and lymphatic biology. So today, Dr. Shawber will speak to us about understanding the genetics of fetal and pediatric lymphatic anomalies. Please join me in welcoming Dr. Shawber.
Thank you, Mike, and for this opportunity to talk to this audience about our recent work and the work over the past couple years that we've had. I chose a very broad topic because I'm going to touch on a couple of things going on in the lab and how they all relate back to really understanding the genetics moving forward. And so let's see, there we go. Just real quick, I have no disclosures, but again, I have been funded, but I really want to point out there are two groups that are here and lead this group, both the UPenn Orphan Disorder Center, this is the $1 Million Bike Ride, and the Lymphatic Malformation Institute pilots have been critical to establishing the research that I'll talk about today. So I'm going to give just a very brief background in genetics and lymphatic anomalies, more where we stand with it, give an update really on the genetics of fetal lymphatic anomalies, because actually the majority of our primary lymphatic anomalies actually present in utero, and it's an understudied topic in our field. Understanding how we can use these genetics to understand and guide therapies, current therapy understandings, and some future work and identification of a new novel therapy in my group. And then bring you up to date on an ARPA-H light project, which is going to hopefully advance our understanding of the genetics of lymphatic anomalies and potential for opportunities in the future to get involved with our group. So genetics, lymphatic anomalies from this audience, you guys understand, are a broad spectrum of different subtypes. They can be focal, such as primary lymphedema or cystic hygroma, or they can be more complex, like central collecting duct lymphatic, it should be malformations, hydrops and generalized lymphatic anomalies. And a lot of the genes that have been identified in these groups, they can be germline, they can be somatic, or they can be a combo of germline and somatic. But I think really to look at this, the big challenge where we're at right now is a lot of the same genes give two different phenotypic presentations. And so this is important because a lot of our therapies are targeted towards these pathogenic variants. And at this point, I would say phenotypic expression is not really predictive of genetic phenotype and best therapeutic responses. So the other challenge that I came across, and because I'm an OBGYN, is in the fetal period, these diseases or subtypes actually have completely distinct names. And so I think one of the goals I've been trying to work on is get commonality of names and recognition of the phenotypes, because this goes into how it's written into charts and those components. So primary lymphedema could be called pedal edema. A cystic lymphatic malformation could be called a cystic hygroma. Central collecting would be idiopathic chylothorax. Hydrops is both, but it's also recognized as generalized lymphatic dysplasia. And for generalized lymphatic anomaly, it's often called prenatally lymphangiomatosis or lymphangioma. However, some of our studies, what we'll talk about, they share the same genes. So currently, I think the questions that we have, and I've updated this from my prior talks, is Why do lymphatic anomalies with identical variants have varied phenotypic presentations and severity? Why do lymphatic anomalies with identical variants have varied therapeutic responses? And can we use genetics to guide our treatment options and identify new ones? And I'll mostly address some of the beginning of the two, but more the third here. So this is currently the genetic diagnostic framework in the postnatal period. And I'm going to point this out because it actually is a long meandering process that I hope with the work of my collaborators that we could speed this process up for our patients, because the earlier treatment of a progressive disease that hopefully will have better outcomes. So the current system is a patient, if they actually get to the proper physician, will have phenotypic assessment, meaning signs and symptoms, what are the symptoms, tissue pathology, and imaging. At this point, they're usually recommended to a geneticist or a genetic pathologist, and the first round of analysis is usually a germline analysis. And sometimes they identify pathogenic variants, and then your pathway is done. But I'm going to show you that the majority of the time our patients don't happen to fall into this category. Actually, what they end up having to do is they're either negative, meaning we have to do another analysis, or the current bioinformatic pipelines, because the way that they're developed based on ClinVar and ClinGen, don't have the majority of lymphatic anomalies genes there. So most groups do not actually query or look at these genes. So the next round of analysis is really doing a deep dive of lymphatic anomaly genes and looking to see if there's pathogenic or variants of uncertain significance. Once you fail this, hopefully we get to having affected tissue. And I think the new thing is affected tissue can be used. And hopefully at that point, because they're somatic, we'll be able to identify what the pathogenic variant or variant of uncertain significance to help guide treatment. But there's a lot that needs to be improved in this process. So the first is, I'm going to talk about studies where we started off querying about 35 lymphatic anomaly genes. And at this point, I'm going to tell you we're at least at 73 genes, and it actually may be up to almost 80 genes from our recent meetings that I've attended. And so just looking at those 35 genes, the other thing that's advanced, and as I said, there's 73 genes we're looking at, but what else is advanced is instead of doing affected tissue, we're now starting to incorporate cell-free DNA. And this could be DNA isolated from plasma or from lymphatic fluids, which are always in much excess in our patients and much more easily accessed. So I'm going to talk a little bit about a study that we published couple of years ago. And in the study, we went back to a group of about 500 cases of fetal lymphatic anomalies studied in a New England Journal paper by my collaborator, Ron Wapner, and Jessica Giordano. In that group, when they looked at this group, it turned out there's a significant number of fetal lymphatic anomalies, but only one pathogenic variant was actually identified in that group. And we thought this was a good category to go back and do that candidate gene approach and look at the 36 genes or genes that we looked at. And so just to show you what these populations look like and how difficult it is to segregate them, we initially had 111 cases that were consistent with the fetal lymphatic anomaly. We had excluded 40 of them because sometimes there's no data. It turns out when we went back to imaging, there was no actual fluid. So this is the challenge of doing any analysis. But there's also complexes and syndromes that cause lymphatic anomalies that are outside what we're interested. Echinacea sequence means lack of fetal movement, which can cause edema from lack of movement. Very complex syndromes where it was not clear that the edema that was associated had anything to do with lymphatics. When you have fetal anemia that precedes lymphatic edema, that also usually is a non-linked twin-twin transfusion syndrome. And there's some chromosomal abnormalities like Turner syndrome. When we broke that down, we had 71 cases, which we called fetal edema cases at this point. And we broke them into two categories because this is also the challenge. 42% of these cases were isolated and resolved during development and disappeared. However, 29% of these cases were very severe and continued on and either led to fetal demise, decisions to terminate, or an affected infant at birth. When we looked at this category, and this is actually one of the first of its kind, what we saw when we looked at this, the significant group and the isolated group that resolved, what really stood out in this component is that diagnosis during that second trimester or late first trimester was much more likely to be a case that's going to go on to be a significant fetal lymphatic anomaly. Those early scans, those first trimester scans, were not actually very informative at all. And you can see that's mostly in the isolated nuchal edema, meaning 93 cases of this isolated nuchal edema, that's the large region that dilates in the back of the neck, actually resolved itself. So other things that were predictive of going on is that more than one chamber of fluid was observed in the fetus. And the other thing is concurrent of other structural anomalies. And you'll see because a lot of these turn out to also have rhasopathies as well. But I think what really stood out, and I actually saw someone report this last week in ISFA that agreed with us, that actually only about a third of the severe lymphatic anomalies or fetal edema actually went on to have a live birth. A third of them had fetal demise, and a third chose termination, which is very different from the other cases. But also, we want to be able to advise parents to make a decision whether termination is really necessary early if we know that it's going to resolve itself and not be a problem. In that clinically significant group, what we also saw that preterm birth was significantly increased in that group compared to the other group. And then the other thing, which I think is actually quite striking, is maternal preeclampsia was significantly higher in this group. And this is important because there is an increased cardiovascular risk to both the mother and the child after birth. So this is actually something that needs to be addressed and understood more. Now, when we looked at the genes, I'm just going to summarize here what we found was this, when we did the candidate gene approach, what we found now, instead of just that one variant in that clinically significant group, we found either causative, which is pathogenic or likely pathogenic variants, or what are called variants of uncertain significance, where we have enough evidence to suggest they'd be pathogenic, but we need more, in about 51% of the cases, suggesting about 50% are germline cases at this point. When we looked at those variants and looked at them, the majority of our pathogenic variants actually were in rhasopathy genes, but we identified about 24%, a quarter of them were variants in PTPN11, RASA1, RAF1, SOS1, and SOS2. And sometimes we had cases that carried two variants, SOS1 and TAI1 was one of them. The other most frequent variants that we found in both pathogenic and variants of uncertain significance were those in piezo1. And this was both in autosomal recessive, meaning two loss of function alleles, but we also saw it with missense variants in a significant component as well, so also autosomal dominant. And I think looking at this, when I advise and get asked about this in the prenatal phase, piezo1 missense mutations or variants are actually much more common in this population, but not appreciated at this time. We also found uses in other genes like CSLR1, EPHB4, and TAI1. What was challenging and why especially the pathogenic variants weren't called, especially in the rhasopathies, is usually one parent was often an asymptomatic carrier. So in a condition where you even have a pathogenic variant in a rhasopathy gene, if the parent is an asymptomatic carrier, and also what happened is also was usually an isolated lymphatic anomaly, there was no return of genetics to the parents because of this misconception that, or variable penetrance that we have with rhasopathy genes, or there's another variant that's giving the penetrance, which we're looking into now. But also what really looked in this is in our cases, They did not have cardiac defects. They really had normal facial features and no short stature or small size. So these are also common phenotypes observed in RASopathies. Again, it was purely lymphatic defects. And I know Sarah Shepard at NICHD also has been observing the same. So I'm going to jump a little bit about that, just saying, really, I think we have to look more closely. There's a lot more lymphatic anomaly genes that we're seeing in germline, and they're just not currently getting called. And so this is something that I'm working on by both joining ClinGen GSEPs to do gene curation, but also want to encourage people to keep publishing on this as well. So the next thing I'd like to talk about is, can we take our preclinical studies if we add genetics to the mix? So can we use genetics to do drug discovery? But what we know already is that drugs are already in use in our patient population. We have sirolimus, alpelisib, and trametinib, which are currently being used mostly off-label at this point in our lymphatic anomalies patients. But I'm also going to talk to you about some preclinical studies where we identified bortezomib, a 20S proteasome inhibitor, as a putative therapy for the future to test. So we see this figure a lot with the group, but really it just points out all the overlapping genes and how they all filter down really to activating two pathways, which is where the drug targeting has happened in the RAS MAP kinase pathway and the PI3 kinase AKT TSC2 mTOR pathway. And this has led to the drugs that I introduced before, and we've talked about sirolimus targeting mTOR, morasidib targeting AKT, alpelisib targeting PIK3CA, and now there's a couple more drugs that are in development that are targeting the pathogenic versions of PIK3CA, or trametinib, or I can't pronounce it ever, but the other version of a MEK inhibitor, a mechanism here to inhibit here. And work done by this group has really started to see how these work, but we also wanted to see, what we hear is a lot of people interchange drugs between these. So do you use sirolimus, everolimus, tacrolimus, and are they really interchangeable between all of these? And also starting to understand how these work at different steps. So to take this approach, what we ended up doing for this is I've been isolating in my group patient cells for probably the past 15 years. And of these cells, because we know they're enriched, we do whole exome sequencing and variant analysis. And we always query for lymphatic anomaly genes in this group and for gene discovery. The beauty of this is we can take the patient cells, and just like patient tissues, we can implant them in mice, let them grow, and they can form the same abnormal lymphatic channels that we see in patients. And this is a system that we can test drugs in, but we can also understand the pathobiology. And then to complement this, we built in the system initially published by Ty McKinnon's group, which is going ahead and expressing the PIK3CA off the ProxCrete ERT promoter. And to do this, we let the mice get to about 3 weeks in age. We treat with 4-OH tamoxifen on the ear, so we generate a mosaic animal. And then we go ahead and we either go out and analyze, or we can start introducing therapies in this. So these are the systems and the cells that we've been working with. So this was done in collaboration with my clinical partner, June Wu, here at Columbia University. And what we did is we took isolated cells that we had whole exome sequenced. In this case, they were cells from venous malformations and lymphatic malformations, because they have a common origin and often an overlap of the genes and pathways affected. And we did a high throughput drug screen where we scanned over 1,300 drugs. And the idea was for rapid transfer, well, as rapid as it can be, to the clinic. So we did drugs that were already approved for use for another use, or were in clinical trials. None of these drugs, well, some of them, I guess, were for lymphatic anomalies, but most were for other use. And so the idea was we could more rapidly repurpose the drugs. And we looked at two different groups. One, we had a group of PI3 kinase, AKT, and mTOR inhibitors. We also did the MAP kinase inhibitors, but I'm not going to show that today. because it doesn't really work by inhibiting growth so much. Looked at this, and really this is a viability assay. So cell death and inhibition of proliferation is our assessment. And another group of drugs, which we were surprised to find were 20S proteasome inhibitors, were actually almost the most effective drugs we found in this assay. So just a quick analysis of really what happens when we looked at all the different categories of the PI3 kinase AKT mTOR pathway. And I think really we're sitting over here, and sirolimus and alpelisib are the two drugs that are currently used in clinic, and they reduce proliferation, but it's usually between 20% and 30%, and that's pretty consistent across the board, no matter what cells we look at. But I think really looking at this, if you look at alpelisib and then you start looking at the other PI3 kinase inhibitors that are out there, there are some that actually have no activity at all in this assay, whereas others like pitilisib actually had almost better consistent activity than others. And we also found, which I'm not going to show, that this was very effective with cells with MAP kinase activation as well. When we looked at AKT inhibitors, there was nothing really consistent across the board with the cells and how they were affected. And mTOR inhibitors, we'd say temsirolimus, everolimus were equally effective with sirolimus, but tacrolimus actually wasn't effective at all. And so it really isn't that these drugs are interchangeable. They have slightly different functions and mechanisms of inhibition. And I think we need to really look at that. And it's something we're putting a publication to discuss currently. But the other thing that came out is that double inhibition of PI3 kinase and mTOR by a drug called omeprazole, but not bortezomib, actually was the most effective at inhibiting growth. Unfortunately, this drug has high toxicity, and all the clinical trials for other conditions, mostly cancer, have been terminated. So this is not a drug that's currently available on the market. So coming back to those 20S proteasome inhibitors, one of the things that we did a prioritization. So the most effective at inhibiting cell growth and inducing cell death versus the least. So sirolimus, alpelisib, and trametinib, they're not horrible because we had 1,300 drugs, so they're still pretty high on our hits. But you saw from the data before, really we're getting about 20 to 30% inhibition of growth or induction of death. However, carfilzomib, dolantamab, and izosumab turned out were always in the top 10, and carfilzomib was actually always the first hit that we had. And we said, what are these drugs? And it turned out they're 20S proteasome inhibitors. And so we actually went ahead and said, what are these? What is the class? And it turns out there's 6 drugs in this class. So it goes from aprocimib, maracimib, isicimib, dolantamab, carfilzomib, and bortezomib. Actually got that out all at one point. And here, the initial screen was 1 dose. This is a dose escalation study, and we compared to alpelisib and sirolimus, and these are the PIK3CA or activation of the PI3 kinase pathway cells that we used. And then we took what would be, it's not the perfect assay, but what would be the clinical plasma dose for the average dose, and figured out how effective at inhibiting growth it was in all of these cells. And when we looked at this, all the 20S proteasome inhibitors were much more effective than these others in this assay. I'm going to go on to talk about certain ones, and I'm going to talk about aprocimib and bortezomib. Aprocimib we started working with because it actually is a 3rd generation proteasome inhibitor that had higher efficacy, longer half-life, and reduced toxicity, and is orally available. But then we came back and started studying bortezomib because it's first in its class, and it turns out this is the only drug that has been used in pediatrics at all in the literature or in clinical trials. And so if we wanted to translate to the clinic, the safety between these two, this has a much better safety information, so hopefully we can bring it faster to the clinic. We also did 2 different doses in a lot of our studies. We did the clinical dose, where you can see there's not a separation with the HD lex so much that are in red, but we also dropped it to 1/10 of a dose. and saw that it was just as effective. And then we started segregating the effects we saw on the control cells. So what is known in the literature, and I'm going to jump between aprocibine and bortezomib at this point with these two drugs, and we've seen very similar responses, is that these drugs have been shown in angiogenesis, no data on lymphangiogenosis, to both reduce proliferation of endothelial cells or angiogenesis in the cancer setting, or induce cell death of cells with proteasome proteostasis defects. So the first thing to look at, we looked at proliferation. And what we saw was, yes, when we did the 10th of the dose or the clinical dose, these two doses, we had a significant inhibition of proliferation, specifically in the cells that were treated, but the ones that were not treated had no effect. The other thing, just to point out real quickly, you see how V-cadherin here is normally at the cell surface and you don't see a lot of cytoplasmic expression. What we always saw was an increase in expression, cytoplasmic expression, and when we quantified it, we actually saw a significant increase with drug treatment. This is V-cadherin in this situation going up. And we think that that's because how proteasome inhibitors work is they work by further exacerbating proteostasis defects. So this also told us that the drugs were working in our situation. But really what we're interested in, and this is what's known in melanoma, is that proteasome inhibitors target cells with these proteostasis defects specifically for apoptosis. And so when we did a tunnel assay, either at the 10th dose or the clinical dose of aprocibine in this case, this is BMECs in this case because we have more data for the BMs, we saw that there was significant death really only in the cells that carry the pathogenic variants. And we saw this also in studies of the lymphatic malformation endothelial cells. So it suggests that bortezomib should target just those pathogenic cells and hopefully will leave the normal cells alone in our cases. So kind of like a cancer therapy. So now we transition this to our mouse models. This is our xenograft model. In this model, we implanted the LMECs expressing PIK3CA variants, let them develop for 2 weeks. And we know from our prior studies and ultrasounds that this is when the channels have begun to develop. And then we did aprocibine in this case in a traditional cancer therapy, which was 2 days on, 5 days off for 4 weeks, allowed it to go another week, and then analyzed. The first thing we saw was a dramatic decrease in these disorganized channels, but also you see how you have these gaps. What we know from our studies is these are cells that have kind of died and sloughed off into this. So even the channels we have much more uniform podoplanin expression and other markers. But when we quantified both lymphatic vessel density, and we also looked at average lumen size, the treatment was significantly reduced abnormal channel development in this model. We then moved it into the transgenic mouse model. This is the ProxPRT PIK3CA H1047, where we induced the phenotype at 3 weeks. After 2 weeks, we started bortezomib treatment in this case. Again, 2 days on, 5 days off. And then at 9 weeks, we euthanized the mice and analyzed the ears. In this model, again, in the wild type or the single transgenics, we get normal vasculature that develops. And here we get a very disorganized, fused vascular plexus, lymphatic plexus that forms. This is live one staining as well. So when we looked at this in the bortezomib treatment, if you look at the vehicle treated in this group versus the just a control ear, we had an intermediate phenotype that we recovered. So we had a significant reduction in the density of the lymphatic plexus that forms in the ear in this. But what else we did is we actually had a better phenotype when we did high magnification. So this has already been shown. The lymphatics in this model are usually in a very active phenotype. They're pointy. They have abnormal V-cadherin expression, almost in a zipper phenotype. when we looked at the bortezomib treatment, what we see, and we think that this is normal cells growing out, and we're still figuring out how to look at the difference between them. But what we think is you have a quiescent bud that's forming, and you start to get the punctate button leaf junctions that should happen here. In the lymphatic plexus, where we were unable to identify any valves in the plexus further behind in the pre-collectors, we started to recover the valves as well in these. So we have very promising data suggesting bortezomib or aprocimab would be a good therapy to transfer over. So just to summarize this portion, not all drugs that target components of the PI3 kinase AKT-mTOR pathway are interchangeable for treatment. But we also identified 20 proteasome inhibitors, especially bortezomib, which we're hoping to get a clinical trial going as a potential therapy for lymphatic anomalies with PI3 kinase activation. Now, I want to talk about how we're going to go to the future and a recent project that was funded by ARPA-H Light about 6 months ago, and give a bit of an update and also talk about opportunities for collaboration in the future with the community. So, where are we with the current status? And it's that we know the genetic causes for about 40 to 80% of primary lymphatic anomalies, or reverse it the other way, depending on the subset. So there's a lot of versions and phenotypes we don't have the gene, and part of it is we're just insufficiently powered when we're all islands unto ourselves. Genetic studies commonly identify variants of uncertain significance that are not always reported back to patients. And this is very specific in the US, what gets reported and not reported. And it's dependent on the site, and it's also dependent on the state because there's state regulations. That current clinical WES and or exome sequencing or genome sequencing for fetal lymphatic anomalies is really geared to germline mutations, and it's insufficient to identify somatic variants. And this is critical because physicians are starting to therapeutically intervene in utero, and it would be nice to have the genetics to support it. And then current US commercial and academic genetic panels only queries about 20 to 60% of the LE genes that we currently know. And so this is where we were at when I put together a group of clinicians and geneticists and researchers to start to address these questions. So, again, why? It's a rare disorder. So most of us are insufficiently powered for gene discovery. Genotype-phenotype correlation is variables. We need to do better deep phenotyping of our patients to understand correlates between genetics and phenotype. And as we're talking, we're expanding from that 36 genes that we looked at 3 years ago to 73 genes, but I just went to a recent meeting and there's at least 3 new genes that will be added hopefully to panels in the future. We also think there's a second hit that's probably influencing phenotype and therapeutic responses, which is also necessitates, we need to be able to look at somatic and germline mutations and be open to that when we're analyzing the data. And then the other challenge is most of these are post-zygotic. And again, just to say, current germline analysis in utero is usually 30 to 50x coverage that you do. And even if you do a panel, it's not that deep either. You need something in the 500 to 1,000 coverage, and these are things we will be working on. So to do this, I brought together a very large group from Columbia University at the top, bioinformaticians who are doing characterizing and doing all of us and coming up with maternal cell-free DNA technology at the Broad Institute, the group at CHOP that has developed the cell-free panel for fluids and lymphatic fluids and plasma when the group, Pia Ostergaard and Sylvia from City St. George's, the Boston Children's Group, NYU and Arkansas. And then also a group, a patient advocate, a therapist, a product development lead, and a genetic counselor who really helps us understand how to do these studies, but also make sure we have impact on our patients. So to do this, what we proposed was actually threefold to do. Number one was to generate a large lymphatic genomic database and collect all lymphatic anomalies, whether they're germline, somatic, all the sequences. And the beauty of the group at the Broad is that they have the technology to harmonize this data and do gene discovery. We will look at it as a giant group. We'll do subgroup analysis. Our goal is over 1,500 unique probands with at least 20, with an aim of 2,000 probands. I'm going to tell you, we've already met our minimal goal, and we still are only 6 months in. We also want to combine this with deep phenotyping. So we're collecting all the information with HPO terms so we can better understand it. As I said, we're using advanced genetic bioinformatics and machine learning to help us look through this data. And we're developing over here, trying to develop a robust method to really figure out how do we detect somatic variants in utero so that we can better diagnose and do early therapeutic interventions, but also bringing together a group to take all those VUSs or genes of uncertain significance to more rapidly screen them so that we can bring it back into this pipeline and have a better expansion. All of this leads to our product that we're developing and hopefully we'll have out by fall of 2027, because that's the goal of ARPA-H and our push, is to develop 2 new genetic panels specifically for lymphatic anomalies that can take blood or lymphatic fluids in combination with the group at CHOP and here at Columbia, that we can take tissue, blood, lymphatic fluids, and do a rapid, high coverage screen to more rapidly identify genetic variants for our patients and increase it. We also are working at trying to get genetics in general more broadly accepted by primary care physicians and general practitioners who don't even know that these tests exist or that our lymphatic anomalies actually are a syndrome. We would like to query germline and somatic variants simultaneously, and I'll show you why that's so important because it changes the time of diagnosis, which is really important for patients. But we're also trying to get an identified genetic testing company, a commercial partner, so that this test will not just be limited to our labs here in Philadelphia or in New York, but that it could be nationally rolled out and supported so that we can have more testing done. So again, just to give you an idea of that current pathway and give you the whole component, is usually if a lymphatic anomaly is analyzed and they can get to a proper center, the first test is usually a germline test. And the turnaround on that is about 2 to 3 months. And depending on what panel you use, it only covers about 10 of the genes. So commonly there's no variant identified, and sometimes they go back and do the germline again. Or if we're lucky, they go and do a second genetic panel and do it at a deeper sequencing so that you can look and find a somatic cause. And again, that leaves us where we are currently. What we'd like to do is increase the number of patients that are actually being recognized as lymphatic anomalies so that they can even get genetic testing, which is our Discovery Duo project. But generate a panel with more genes, more comprehensive, that can use all the commonly generated samples. You can do it as a prenatal or postnatal test. And it's designed and regulatory approach that we've taken is that we can add genes to this panel as they come online. And our goal is to at least go to 70% of LAs have a genetic diagnosis, but really we'd like to have almost all of them at the end. So just to see where we're at, this was actually 2 months ago, but just right now we have about over 1,700 unique probands from all of these sites. We're doing our first harmonization and analysis of our pipeline in the coming months. So hopefully we'll have new genes coming out. We also are using public databases and fetal consortium data to increase this number so that we're not biased. And I would say, hopefully, we're open to additional collaborators in winter or spring of 2027. So please reach out to me if you'd like to be involved in this study, meaning we can add your samples to increase the power of our database. The next is to detect somatic variants. basically it's been shown once or twice in like cloves that if you do deep sequencing, you can detect um, somatic mutations. And we even had a recent case where a baby was born that had 50% somatic PIK3CA, and there we could also detect it in a traditional CBS. No, it was amniocentesis in that case. So it is possible to do, it's just not standard protocol. And so why is this important? Because there are now reports, and we heard a bunch of them last week at ISFA, that clinicians are using rapamycin or sirolimus and getting good outcomes for cervical lymphatic malformations. However, if 20% of these cases don't have a PIK3CA mutation, are we exposing them to a drug in utero without knowing what the long-term effects are on development and other parts of the child's life? So I think we need to be able to genetically diagnose these somatic cases as well. So we are currently open and we can do virtual recruitment. So if anyone has these cases, we can, and you're interested, we would be very interested. Basically, we're very open, but not limited to these diagnoses here that we have and we shared here with this. But we are excluding from our prior studies those first trimester findings with just isolated increased nuchal translucency, isolated first trimester cystic hygroma that's not observed on the second, and suspected confirmed aneuploidy. And again, echinacea, fetal anemia preceding the edema, and twin-twin transfusion. What does the study involve? Again, we can consent virtually using REDCap at any site if you're interested, but really we're not doing any excess interventions. It's excess amniocytes or DNA, excess DNA from amniocytes that you have. We're planning on doing like 1500x coverage sequencing to see if we can detect, and then 2 10 ml Streck tubes of maternal blood. And this is to see, can we detect it in cell-free to have a truly non-invasive test? And these are going to be read at much higher coverage. We're not doing any results are not being returned and no payments involved, but it is something that we are very interested in increasing our recruitment for. And then I'd just like to acknowledge my group here. This is us last week in Philadelphia at the ISFA meeting, who is really critical for the studies here, but also everybody else in RED who's really contributed to this and the whole group. And I'd like to also recognize my new ARPA-H component and the new data hopefully we'll be generating in the near future, and all of our funding source. And again, remembering LMI and the LMI LGDA $1 million bike ride have been critical to funding our studies. And I'll thank you there.
A great presentation, Carrie. I'll go ahead and get the discussion started. It seems like the RAS pathway-driven LMs would also be sensitive to the proteasome inhibitors. Have you looked at that at all?
Yeah. Yes, we have. There's just a lot of work to do, and we proposed a grant to do it. Our initial studies, like actually the cells that are behind me, PTPN11 cells also have AKT, and we do see that the proteasome inhibitors actually work very well with them, and they also have these proteostasis defects that we think that they're being targeted. So I think because of this crosstalk, it's definitely going to, the RASopathies will also be responsive. The clinical trial that we've been trying to develop, and we're talking to 2 different groups, hopefully to get funding to do it, is we're going to do a basket trial because it's really rare. And one of the baskets is going to be these complex lymphatic anomalies that are RASopathies. And then we'll also have more like the PIK3CA lymphatic malformations, and then the venous malformation bucket is what we plan to do. So I would predict, I mean, we still need to do the experiments and the mouse line and the in xenograft models, but I would predict they'll also be responsive. The xenografts are harder with the RASopathies because they're not as proliferative as the PIK3CA ones. So there's something compensating between, even though they have the PIK3CA, they're just not quite as proliferative. And I think your work with the valves really starts to explain why they're different.
I just have one more question to keep the discussion going, and this is with the PIK3CA driven LMs. Sort of my thinking of this is that you need this hyper pediatric kinase AKT-mTOR signaling to be driving really this translation. And the more translation that you have in the cell, likely the more misfolded proteins and abnormal proteins you'll create, resulting in the dependency on this proteasome.
Yeah.
Uh, so have you looked, does like treating with rapamycin, would that not be a good combination to
Yeah.
include?
So
Because
it's a good question. You've seen some of my postdocs talks.
Mhm.
Um, we do think what's happening is the transcription and translation that's leading to increased V-cadherin and CD31. And in LM cells, we actually see that that leads to a lack of it getting to the cell surface. And so we're not and if you see patient cells, you'll see these breaks and everything and a very discontinuous endothelium. But we think what's happening, I'm just going to jump back. That quiesces the cells, right? So that's the quiescence. Having V-cadherin out and CD31. We have done sirolimus, and it does exactly what you said. What it does is it brings down the protein expression. We still need more cells, so that's why we're not showing it yet. But it brings down the protein expression. We get much more V-cadherin and CD31 to the cell surface and much better junctions. And we think that's what's leading to the suppression of proliferation. So it may explain why in about 40% of cases, when you remove sirolimus or you do it too soon, you get a rebound or you have an effect because they're no longer that, the V coherence not there to quiesce anymore. So coming back, I know, to be honest, we did propose in our grant to look at sirolimus and these proteasome inhibitors simultaneously, but I think they're going to be opposing each other.
Mhm.
So probably not the route to go.
And if you have a question, just feel free to unmute yourself and ask your question, or if you want to raise your hand, I can call on you.
I have a question, a clinical question. Do you know what the side effects are of proteasome inhibitors in multiple myeloma?
Yes, and we actually, my clinical partner and I, June, and I just recently published a lot of the side effects for bortezomib in particular, in Frontiers of Pharmacology, doing a review. So in adults, the major side effect is peripheral neuropathy, and then a bit of an effect on your hematopoietic cells and your myeloid cells because they're so proliferative. When we looked at in pediatrics, really that fell out. The peripheral neuropathy is not really experienced in pediatric patients, but they do have issues with monocyte cells depression. And so infection is a major concern. And so even in those cases for children, it's usually blood cancers in children that are being treated with bortezomib, is to also treat with low levels of antibiotics to prevent the sepsis or infections from occurring. So you're basically immunosuppression, kind of like sirolimus, unfortunately, happens. But some of the cardiac and the neuro effects are not really observed in pediatrics. And that's not unusual for drugs. There's pediatrics have different adverse event profiles to the same drugs as adults.
Thank you. I have another question. I can put my video on too. So I was wondering if, for alpha-7 versus we have a biomarker, which is the genetic mutation to some degree. But
Yeah.
for bortezomib, do you have something that you like? Who would be when you're thinking about actually doing a clinical trial? Who would be in your cohort that you would focus
As I
on?
said, we're going to do a couple of different buckets, and one will be lymphatic malformations with PIK3CA variants because those will be more common. We're also going to have a bucket with venous malformations because we also saw that we get good responses in TEC mutant cells as well as the PIK3CA cells. And then we have a group that's just going to be complex lymphatic anomalies, and we're not going to give the genotype at that point. We figure a lot of them are RASopathies, but we don't want to limit that population because it's a very small population coming in. Advice from Denise Adams when we were designing this trial.
Sorry, just to clarify, I mean, like, why would I, as a clinician, choose to put someone on that trial rather than do
Then
treatment with?
alpelisib,
Yeah.
because I hope what we're going to show is going to work like a cancer therapy and we're going to destroy the cells. And so I think alpelisib and sirolimus, and this is like purely hypothetical. We're still figuring this out. We need to look at it in our model. But what it looks like is that we're targeting cells with the pathogenic variant to die. And so hopefully we can actually, if we can get early enough intervention, just eliminate those cells and leave behind the normal wild type cells to come in and fulfill. Does that mean we probably will not be able to use this in germline cases? I don't think it's going to be a drug. So like for say RASopathies, MEK inhibitors and mechanists are looking really, really great in that population. I don't think we'll be able to do bortezomib if it's going to kill off all the cells in the patient. So these are things that we've been thinking about. We're hoping it will be just one treatment instead of a lifelong. So maybe it's one course of 6 weeks, and then hopefully you're off the drug. So cost-wise, hopefully it comes down. That's our thinking, at least. We have to prove it still.
Thank you.
Well, thank you so much, Carrie, for a great presentation. And we'll see everyone next month for our next presentation.
Yes.
All
Thank
right.
you. Thank you for the invitation.
Bye-bye.
Bye-bye.